Hello, my name is Cláudio Pinheiro, and together with my colleagues in the Experimental Cancer Research Lab, I study the importance of the protein corona surrounding cancer extracellular vesicles. Communication is something extremely natural and important in our day-to-day lives, as we interact with other people. This is no different for the cells in our body. All cells in our body are capable of communicating in short and long distances by releasing tiny bubbles that they use to communicate with each other in the body. These bubbles are called extracellular vesicles. You can compare them to letters or little packages filled with information. This information can be transported from one cell to another and can then produce effects in the cell that receives it. Extracellular vesicles can be found in all bodily fluids, such as blood, urine, and even tears. As such, these little letters are the way that all of our cells communicate with each other, including as well the cancer cells. This type of communication between cancer cells has shown to be the key in cancer progression and spread, as these vesicles can, for example, already create a cancer-friendly environment in a distant organ, as such paving the way for later cancer metastasis. Recently, we discovered that extracellular vesicles contain components around them that can, for example, indicate their destination. Think of it as stamps and postal codes in letters. If the wrong stamp or postal code is placed on a letter, it will never arrive at its destination. When it comes to extracellular vesicles, these stamps take the shape of a protein corona, which are several proteins that surround the extracellular vesicles and have important communication functions. We know that such protein corona is also present around extracellular vesicles released by cancer cells, but we don't know yet how this corona looks and how it is involved in cancer progression and spread. Extracellular vesicles are extremely small particles that are found in the human body, and to study them is no easy task. In our laboratory, we recently acquired Flanders' first super-resolution microscope. This cutting-edge technology allows us to see these extracellular vesicles with unprecedented detail and their respective protein corona. By using this super-resolution microscope to visualize extracellular vesicles present in blood plasma of cancer patients and healthy donors, we hope to be able to characterize the unique protein corona present around cancer extracellular vesicles. Once mapped out, we aim to investigate how this protein corona affects cancer progression and how we can adapt or modify it to use it to our advantage. There are several ways in which a better knowledge of cancer extracellular vesicles can impact cancer diagnosis and treatment. To start, if we can identify a unique protein on the corona of cancer extracellular vesicles, this can be used as a biomarker for the early detection of that cancer type. This means that if this unique protein is found in someone's blood, we know it means there's a cancer tumor growing somewhere. Alternatively, if the presence of a certain protein is a true cancer-targeting stamp, directly sending extracellular vesicles to a distant cancer cell, we can think of targeted delivery of therapeutics to cancer cells, using a specifically designated vesicle with the correct stamp or corona. Overall, we are convinced that the completion of this project will allow us to understand the utility of the proteins surrounding extracellular vesicles and how to better use them to our advantage, unlocking a full range of potential new applications and benefits for the treatment and detection of cancer.